Om. Good afternoon, everyone. I'm very thankful to be here in a large lecture hall and being blessed by the chairperson, whom I humbly regards very much. Sir Pradam to both of you. I appreciate this opportunity and I thank the organizers to give me a time window to speak about the thoughts that we have and the type of research we have been conducting at Patanjali. Today, I'm going to focus a little bit more about new drug discovery. And when I say new drug discovery, that implies that we're going to talk about how the drugs are being discovered and how do you do in Ayurveda, that becomes a challenge too. So either we can find out the new medicinal plant, which has a capability to treat, cure, or heal the disease, or we can look at the existing plants and look for the plant parts which have not been explored in past. And the good example for that is Ashwagandha. So today I'm going to talk about Ashwagandha at length and try to convince you that these medicinal plants, these wonderful drugs do have a tendency to take care of the diseases which seems really, really difficult to cure by a synthetic route or by the route which have been taken up in the past as well. So I'll start my presentation, and we are blessed by Param Pujya Swamiji, Param Shraddha Acharya, and I go in their holy feet that their guidance and inspiration has been the force for whole Patanjali Research Foundation to actually take it forward. So just a brief couple of slide introduction for those of you who are seeing it for the first time, that Patanjali Research Foundation was inaugurated by our Prime Minister Shri Narendra Modi ji in 2017, and since then it has been a center in its infancy and has been doing a fantastic science since then. These are probably one of the only kind, and we believe the uniqueness in terms of the composition of technologies, equipments, and the scientific willpower that PRF exists, that takes the research operations on a daily basis. And we get inspired by our two great gurus, and we get to do things, we get to play with the toys and the tools that we have with us. Now, these are just a glimpse of the infrastructure that we have for Ayurvedic Research Institute. What you see over here, they are literally pharma grade science. What you see in a pharmacology or a pharmaceutical industry, the similar kind of things exist here at Patanjali Research Foundation. And we have various sections. We do in vitro biology, that means the research with cells. We do in vivo biology. We do a experimental part with animals. We do clinical research. We do microbiology. And most importantly, we do a very detailed herbal chemistry, because that is the one area, that's the one part where we have to show the type of drugs we have been working with, what they really contained. So the detailed chemical characterizations of the phytocompounds which have been packed by nature in these drugs becomes a center of attraction for us and for everyone else around. Now, so today I'm going to focus on Ashwagandha, and as I said, that the new drug discovery in Ayurveda with scientific evidences. And since I'm telling a story, every story has a hero. Today's hero is Ashwagandha. I'm going to talk about it. So in Ayurveda, we know for the fact that, the part of the plant, which is root, where my cursor is right now moving, have been recommended in the ancient classical text, that which have been there for the various treatment modalities. But how about the whole plant? How about the flowers? How about the fruits, seeds, and the leaves? Does these plant part also has something which is unique to them? So with that question, we started to hypothesize and started to look what contains in there. So we start working on the seeds. We are very happy to report, and I'm very proud for the fact that Patanjali Research Foundation is the pioneer institute where we have done the first research on the seeds of Ashwagandha and found that if you use supercritical extraction, which is based on liquid carbon dioxide, you can get some oil. And these oil, which is what we call WSSO, Vidania somnifera seed oil, it has a very strong anti-psoriatic effect, antimicrobial effect, anti-biofilm capability against salmonella, and also has an anti-candidiatic effect against candida species. And I'm going to show you some data on that. For whole plant, we have taken the extract and worked on various specific parts. During COVID, we have worked for the SARS-CoV-2 interaction thing. And I'm going to show some data there as well, and can also show it contains other medicinal value. For the leaves, this is the recent finding we have got. We found that the leaves of Ashwagandha plant has a component which has a very strong anti-obesity effect. It controls the fat regeneration, it controls the lipolysis and lipogenesis and the ratio between them. And from the roots, everyone knows that they do have a fantastic capability. Our team led by Dr. Kunal Bhattacharya, who's now in the audience, has worked on quantum dots. We made a very nice little tiny particles from ashwagandha root extract and could show that they have a huge capability on wound healing. So first I'm going to talk about the oil, which come from the seed oil. So we took this technology, which is called supercritical fluid extraction, where we use carbon dioxide as a solvent to take the oil out, and that oil has fantastic capability. So if you see that this is the detailed chemistry, the one I'm talking about, we take a pride in analytical chemistry capability that we have at PRI, and you can see the oil that come from ashwagandha seeds contains such good quality fatty acids, which is expected from a plant-based oil. But what it does that you can actually do a detailed experiment. This is the data from an animal experiment where the psoriasis was induced on the ears, and the animals were fed with the oil, and they were also applied topically with this oil. And as you can see, from the normal animals to a diseased animal, and when you apply the oil, you see these are the changes that happen in those animals, and we could see a very strong anti-psoriatic effect of this oil, which got published in Biomolecules like 3 years back, and we were first one to show that these fatty acids are indeed having a capability. And then, as I was saying, that we also look for the ashwagandha seed oil against a specific pathogen, and we could show by doing our microbiology led by Dr. Swati Haldar that these oils do have a capability to control the growth of the bacteria, the mortality of the bacteria, its swimming and swarming, and at the same time can also control the total path of its replication. That means it can actually disrupt the wall of the bacteria, and bacteria could actually can die. So this is a very strong dose dependence data that we happen to see from the same oil. What is more important for the bacterial research, that all bacteria do have a capability to generate biofilms, which becomes very challenging to deal with. Most of the modern antibiotics have a difficulty to break this out. So we took this task and started to look for the generation of biofilm, and you can see when you give ashwagandha seed oil, the maturation of the biofilm get inhibited, the initiation get delayed, and at the same time, when you look at the mature biofilm, if you look for these findings, you can see these red marks shows a single bacteria. This is a solid biofilm, and as you treat with ashwagandha seed oil, these biofilms start to break down, and this got published as well. Now, the third part, which is very critical for Candida. We came out of one pandemic that was due to COVID, and the next pandemic, what is being forecasted, is the one that's going to happen probably because of Candida. This is the world map in 2024. This was a little bit of geography where the Candida auris infection was there. This happened in 2014, this happened in 2017, and this is 2018. As you see, the time is passing by, the whole world is actually going to grapple too much with this condition. That means the next trouble that we might get would be the candidiasis. So we deal with this, we started to look for that, and then we look for the antifungal capability, and we got a very specific strain of the this Candida, which has a resistance for a known antifungal agent. So if you can see, this is the untreated fungal colony. This is treated with the fungal agent, and you can see this fungal agent cannot kill the bacteria or this fungus. But if you treat with ashwagandha seed oil, the total amount of white dots you see, they get to decrease, which is as comparable to amphotericin B, which is one more antifungal drug. So you see this is FLC, which has no impact. This is amphotericin B, which has an impact, and this is ashwagandha seed oil, which seems to actually control this drug-resistant C. aureus. We then looked ahead for various clinical isolate that we taken from various parts of the world, from Taiwan, Italy, Turkey, US, Australia, and various parts of India. All of these clinical isolates were taken, and we tested this and found, yes, indeed, ashwagandha seed oil, WSSO, has a capability to actually take care of this fungal growth. So this actually got very exciting, and then we find that why that is happening, the mode of action that our gurus always say that we need to find a karyakari siddhant. That means you should know how the drugs are working, which is the one area where Ayurveda have been putting a lot of focus, and PRI and PRF have been spearing it that we try to find out why that is happening. And we found that, yes, indeed, that the various part of the permeability that is being affected by the seed oil. Now, this fungus also generate biofilm. So we looked at those biofilm formations, and if you can see, these are the scanning electron microscopy data, that this is the biofilm under SEM, which is very tight and condensed, and giving two specific antifungal disease, antifungal drugs, FLC or AMB, doesn't have any impact on them. But when you treat them with ashwagandha seed oil, you can see this biofilm start to break down, which is really fantastic. And we are very happy to see this, that this happened at the concentration which are biologically more relevant. And this we then take it up and got it published in different microbiological journals. Now let's move towards the whole plant extract, because we talk about the seed so far. Now we go towards the whole plant. What does the whole plant contain? So we actually did a lot of research during COVID time, and we found that whole plant, if you extract it by using a basic hydromethanolic extraction, you get various compounds. And the compound that we focused on called vidinone, which is there in the root as well, but somehow the leaves has a very high amount. So we could generate an vidinone-enriched Vidania somnifera extract. In short, we call it wine-wise. And this wine-wise extract has a fantastic capability of doing these parameters. So I'm going to show a little bit of data that we did for the first time there during the COVID time. I think this research happened in 2019 or so, and we got it published in 2021, that we found that Vidania somnifera, if you do a molecular simulation experiment, does indeed affect the interaction between the virus protein, which is coming from here, from spike protein called receptor binding domain, and with the ACE2, which is our host protein in our cells. This interaction can be blocked by vidinone. We did by binding, and then we confirmed it by doing biochemistry, and this biochemistry was done in a cell-free manner where we could take both of those proteins and see how their interaction happening, and that interaction can be inhibited by this extract in a very, very low dose. That means the affinity of that interaction was actually pretty good. With the IC50 of 0.33 nanogram per microliter is actually very good dose for a herbal extract. We took it forward, and then we did some experiment, and this is just to show the chemistry. As I said, that chemistry becomes a core for us, and we try to see what each test article might contain, and we could see that this extract has a good sizable amount of vidinone. And then we did the experiment on zebrafish. So during the COVID time, it was very difficult to actually set up an in vivo model for the viral disease. And given the fact that rats and mice do not express the ACE2 receptor, that means that virus cannot infect them. And during the lockdown time, it was even more difficult. So what we do, and that was a big challenge as well, that we worked with a zebrafish. Zebrafish has a swim bladder, which is an analogous organ to our lungs. It has an air sac inside it. So we took human lung cells, A549 cells, and injected them into that air sac and hoped that whether these cells can be deposited inside there and can they multiply. So we injected and wait for 7 days and found, yes, indeed, these human lung cells home in to the area of the swim bladder of the zebrafish. That means now these fish become humanized zebrafish model. And there then we injected them with the SARS-CoV-2 recombinant protein. So this protein would going to induce the same kind of inflammatory response that would happen hopefully in human. And then we look for the various endpoints and try to measure if we give ashwagandha whole plant extract or the enriched extract that has a vidinone, what kind of responses happen. We measure the behavior, this behavior in terms of the fever, and that you can see the behavioral fever, because the fish being a cold-blooded animal, it is very hard to measure the fever, but if you keep them in a chamber which has a different temperature and allow fish to swim across, you can then see how much time they spend in each chamber. That would indicate what's their body temperature. So we could see very easily in normal condition, fish spend more time at that temperature. In disease condition where they were induced with the SARS-CoV-2 virus spike protein, they went to a higher temperature, suggesting that they are going under hyperthermia. That means they're developing fever. And when you treat them with the wine-wise, you can see there is a nice recovery and they tend to shift it out. You can quantify this putting a camera on the top and measure for 3 minutes and actually do a video recording and calculate what's the total time given over there. That indicate, yes, indeed, this drug has a capability to take care of the inflammation induced by SARS-CoV-2 virus spike protein. We measure various other parameters, and I'll be very happy to talk about it if any of you have any query. And this got published in Drug Design, Development, and Therapy, where we could show that from ashwagandha, vidinone could actually attenuate SARS-CoV-2 and host ACE2 interaction in a humanized fish model. Now, let's come to the next part, which is looking for sporothrix. So our microbiology team is very strong, and they were very happy to test this extract into a specific fungus which causes other kind of diseases. And you can see as you increase the dose of this extract, the total fungus growth was also decreased, and that actually shows very clearly how that works out. We got it published in Pelos or neglected topical disease because this fungus is a part of that class which causes these kind of phenotypes. Now, this is the last data that I'm going to show, and it's very latest one and very exciting, that we were actually very happy to see that what really happens in the leaves. Acharya and Swamiji says that people who are obese, someone like me, who take these leaves, they would be able to get some benefit. So I started using them for myself too, and I'm reporting some benefits. But we also did some experiment with the animals and with the cells, and try to find that is there a specific anti-obesity impact of the leaves of ashwagandha. There were some thoughts that if you are taking root of ashwagandha, that is only called ashwagandha. Everything else would be called something else. And there were some advisory from the government and from regulatory agencies. So we actually try to find out what really happens and which part of ashwagandha we need to get focused on. So data what you see right now for Vidania somnifera leaf extract, which we did, and I'll just show you some data. So these are the ones that we did with the cells. These are 3 cells which has a tendency to accumulate fat. And if you label them with a dye that actually that labels fat red, you can see it here. For example, here, if you see these are all the fat is actually there, which is in form of the red thing. If you start treating them with this ashwagandha leaf extract, WSLE, ashwagandha leaf extract, you would see as the dose going up, the redness is going down, and you can see what's the lipid accumulation. This is from the well directly. You can see, you can quantify them and see very nice dose-dependent decrease into the lipid accumulation. At the same time, this extract has no adverse effect on the cell life. You can see what's the cell viability, and up to the dose that we tested, there was actually no adverse effect. Now, this happened because of the various transcription factor changes, various gene expression. I'm not going into detail for the interest of time, but you can see that various gene expressions, which are the critical factor for fat metabolism, that are being controlled by ashwagandha leaf extract. We can then go down to Western blot and look for the various protein, whether it's looking for PPR gamma or CB beta, that you can see those changes do happen. You can normalize and find, yes, indeed, this extract has that impact. And then we switched towards C. elegans that have been our model of choice for last 2 years, which is a small nematode, something like an earthworm, where we can develop various disease model. The life type of C. elegans is 21 days, and they are transparent, so a lot of things can be done with this worm. So there we can do experiments with various mutants. We have close to 110 different type of mutants which have been labeled with dyes or different GFP proteins. So they become a very good marker for a disease measurement. So here, if you see, on the top, what you're seeing is just a fat metabolism by stirring them with oil red O, which label them with the red color. So again, the way you see on the cells, similarly happen in the worms. As you increase the dose of WSLE, it goes down. And then these are the various mutant where the FAT6, FAT5, and FAT7, the genes which regulate the fat metabolism, they have been labeled with GFP. You see the bright green color, that means the expression of this gene is very high. And as you treat them with WSLE, these signals are going down, indicating that we are going in the right path. So this data have been published last month in International Journal of Biological Macromolecules, and we could show the pathway that goes through by playing with the MAP kinase signaling angle. So last slide that I want to show, which is very critical, this I'm going to skip because Dr. Kunal has already talked about it, which is looking for the nanoparticle synthesis and making quantum dots on it. But what I want to come on this is this part, because we are saying about that ashwagandha root are the good drugs, but someone is saying the ashwagandha leaves may not be used. So then we look for whether these drug has any adverse effect. So to measure that adverse effect, we conducted GLP tox studies in rats up to 1,000 milligram per kg per day, and we keep them on a daily basis, and this GLP tox tox data have been published in scientific reports where we showed that if you take these leaf extract up to such a high dose, you do not see any adverse effect. This is to show their body weight gains, their feed consumption. At the same time, 33 different organs which have been harvested, and histopathology was done, all the biochemical assessment done, all the clinical chemistry was done, and we could find that the no effect adverse and no observed adverse effect load dose, which is called NOEL, for Bidhania somnifera whole plant, and the leaf extract was 1,000 milligram per kg. Up to at that dose, there was no change that we found. So that would mean that you can take this part of the plant, you can take almost every part of the Ashwagandha plant, and they could be a potential drug. And that's a new drug discovery I'm talking about. That means that in place of finding a new plants with a medicinal benefit, you can also look for the existing plant and trying to find any part of there which has not been explored earlier can be taken up for the drug discovery and for the human health and taking care for the etiologies that we have been dealing with. So that comes to the last slide of my presentation, which is the motto of Patanjali Research Foundation, where we strongly believe Sarve Bhavantu Sukhna, Sarve Santu Niramaya. That means the science of medicine, from the oldest medicinal knowledge of the world, we offer to the whole world, and everyone remain happy, healthy, and fit. Thank you very much. You have been a fantastic audience. If there are queries, I'll be very happy to take them up.